So, continuing with another episode of As the Stomach Churns, let's continue with our journey through the stomach. So, now that the contents have moved past the esophagus and into the stomach, there's going to be quite a bit of physical mixing going back and forth in here. But this is going to be coordinated by some hormones that we're going to see that will be secreted not only by the stomach, but by other organs of the gastrointestinal tract. And we'll also see there's a lot of enzymatic activity here. So let's delve into that. We've already talked a little bit about the external part of the stomach, the 3 layers of smooth muscle that we have, including that oblique layer that's closest to the lumen. Then we have our circular layer and then our longitudinal layer. We talked about the fact that our epithelium, our simple columnar epithelium there, is a secretory sheet. So it's going to be producing lots of mucus. Without the mucus, the stomach would pretty much eat itself up or burn itself up with hydrochloric acid. So we've got these gastric pits, and these gastric pits are shallow depressions or infoldings of the mucosa, and they will actually then form gastric glands below that. And so we're going to see that at the neck of the gastric pits, we're going to have a lot of mucus cells that are making lots of mucus. We're also going to have some stem cells within these regions that are going to be producing more epithelial cells because, as we've seen, these cells will lead a hard life. They'll need replacing often. So let's talk a little bit about the gastric glands. So we have gastric glands all throughout the stomach, and they all are going to secrete something a little bit different in terms of the main products that they secrete. So the fundus in the body of the stomach, we're going to have a slightly different even histology of these glands as we move to the pylorus of the stomach. But we're going to see that the gastric glands will contain several types of cells, one of which is the parietal cells. And the parietal cells will be secreting hydrochloric acid, and they will also be secreting intrinsic factor, which is going to allow for the absorption of vitamin B12 later on when we get to the small intestine, actually especially when we get to the ileum, which is the latter part or the last part of the small intestine. This is where we will see most of our vitamin B12 absorption, and if we did not have that intrinsic factor, then we would not be able to absorb the B12, and we could end up with a condition called pernicious anemia, and this is where we can't make red blood cells. So this is a problem. So obviously we want to be able to make sure that we're secreting our intrinsic factor, and that's going to be done by our parietal cells. Then we have our chief cells. And as the name implies, this is the chief of what we think of the stomach doing. It's going to secrete enzymes. So the chief cells will secrete a proenzyme called pepsinogen, which when it reaches the lumen of the stomach and reaches this low pH, that proenzyme will start to denature. It'll unfold itself, and then it'll actually have an autocleavage event where it cleaves off part of it, kind of like pulling the pin out of a grenade, and now that enzyme pepsinogen will now become pepsin and is now active and ready to get to work on proteins. The reason that the chief cells secrete it as pepsinogen is obviously we don't want enzymes active inside the cells, otherwise the cells would eat themselves up. So they secrete this proenzyme, pepsinogen, which then is converted to pepsin when it reaches that low pH environment of the lumen of the stomach. We also have a gastric lipase that's secreted in the stomach, although it's not nearly as probably important as other lipases. So yes, the stomach does make some gastric lipase, but more importantly, it's going to be making hydrochloric acid and pepsin. All right, so as we've said, the parietal cells are going to secrete our intrinsic factor and our hydrochloric acid. Our chief cells will be secreting both pepsinogen and gastric lipase. Pepsinogen, as we've said, is that inactive enzyme that, just like pulling the pin on the grenade, as we've said, has to be activated before it can get to work because we don't want it active inside the cell. Interestingly, with the parietal cells, they secrete hydrochloric acid, but they don't secrete hydrochloric acid. They don't make it inside the cell. What they actually do is they will take carbon dioxide and water, combine it to form carbonic acid, and then that carbonic acid will dissociate into bicarbonate ion and hydrogen. The hydrogen then will be pumped out into the lumen of the stomach, and then that bicarbonate ion will be exchanged through a transporter at the opposite surface. So the hydrogen ion would be pumped out at the luminal surface, at the apical surface of the cell into the lumen of the stomach. And in the basilar surface, there'll be a transporter that exchanges bicarbonate ion for chloride ion. And then that chloride ion will be brought into the cell, transported to the other side, and then pumped into the lumen of the stomach. And it is there, inside the stomach, that the hydrogen and the chloride ion will combine or be hydrochloric acid. If these parietal cells made the hydrochloric acid in the parietal cell, again, they would just burn themselves up. So both of these cells have mechanisms so that the toxic or caustic things that they are producing aren't toxic to them and aren't going to be problematic. They would be toxic if they were produced inside the cells, but instead those things get put together, so to speak. In the case of the hydrochloric acid, it gets put together in the lumen of the stomach, and in the case of the pepsin, it's only activated in the lumen of the stomach. It is actually secreted as this pepsinogen. All right, so here's some pictures of some gastric pits, and here we see sort of the neck here, and here we see our glands here, and we're going to have our parietal cells and our chief cells sort of interspersed throughout these glands. We're also going to have some other cells that we're going to find in here as well. So if we zoom in on one, and we can see lots of mucus cells here. We can see our parietal cells colored in this orange color. We've got some chief cells, which are this pink color. We've got G cells, and G cells are gastrin producing cells. So gastrin is a hormone. And gastrin will be produced by the stomach as well as some other downstream parts of the gastrointestinal tract. And gastrin increases the motility of the stomach, and it also increases the secretion of hydrochloric acid and pepsin. So this is a way that the stomach can actually stimulate itself. We also have another type of cell in here called the D cell, which isn't shown. And that is going to secrete a hormone called somatostatin, which will have sort of the reverse effect or the inverse effect of gastrin. All right, so by the time we get to the pylorus, we're not secreting as much enzymes and not quite as much hydrochloric acid. Now we're shifting the focus more toward mucus. So those enzymes that are secreted sort of toward the fundus and the cardia, that area of the stomach where the food will be, that bolus will be received from the esophagus. These will secrete a lot of our enzymes and our hydrochloric acid. As we move toward the pyloric antrum and the pylorus, we're going to shift our focus to our gastrin, somatostatin, and mucus. Lots of mucus, because remember, we've got a really low pH in the stomach. And the stomach is going to be moving, churning back and forth, and it's going to be mixing the contents. And so we've got these gastric juices, which are a combination of all the things that the stomach is secreting that are mixed in with the bolus, and it makes this soupy stuff called chyme. That's C-H-Y-M-E, chyme. And that's this soupy stuff in the stomach. Let's talk a little bit now about how gastric activity is regulated. We have three layers of control here. We have the central nervous system, specifically the parasympathetic division of the autonomic nervous system, which mediates our rest and digest functions. That will be working with the enteric nervous system, which is that standalone nervous system that's contained within the mucosal and enteric plexi between the muscularis mucosa and the muscularis externa. And that's going to, by itself, be able to control a lot of the local activity within certain regions of the digestive tract at any given time. And we'll see this is true within the stomach as well. We'll also see hormones that are produced by the digestive tract that act on either the same organ that produced it or other organs within the digestive tract. We've already seen two of those hormones. We have seen gastrin, which is going to increase the production of hydrochloric acid and enzymes, as well as increase stomach motility. We also have seen somatostatin, which has sort of the inverse effect, and then it will decrease motility and decrease the production of enzymes and hydrochloric acid. Now let's talk about the three phases of gastric control. And the first of which, but not the least of which, is the cephalic phase. So it all starts in your head. Now I'm on the seafood diet, as you know. I see food, I eat food. So as you're thinking about food and contemplating it, maybe you smell it, maybe your neighbor has some really tasty food, maybe as we have in class, someone's crinkling one of those Dorito chip bags. That's going to make you think of food. So often if you're hungry, then you're going to start thinking about food yourself and consuming it. So your brain is going to prepare your stomach to get ready to take on food. And if you recall, one of the major output pathways of the parasympathetic nervous system is the vagus nerves. So the vagus nerves are going to be responsible for approximately 75% of parasympathetic outflow. You'll get stimulation via the vagus nerve, and that's going to stimulate our submucosal plexus. We're going to have mucus cells that are going to start secreting mucus. We're going to stimulate the chief cells to produce pepsinogen, and we're going to stimulate the parietal cells to start producing hydrochloric acid. And once the pH of the stomach gets low enough, then the pepsinogen will be converted into pepsin, which is the active proteolytic enzyme. At the same time, we're going to stimulate G cells to produce gastrin, which will start those waves of motility, start some peristalsis in the stomach to get it to start mixing things. And this is what you feel when your stomach growls. So when you're thinking about food, and now your stomach's preparing for the digestion of this food, then it's sending a signal back to you, hey, feed me. So when you do put food in the stomach, then that initiates the second part, the gastric phase. And this is when we have stretch receptors, baroreceptors, chemoreceptors in the stomach. The stretch receptors will be sensitive to the fact that the walls of the stomach will be distending as it's accommodating the stuff that you're putting in it. We're going to have chemoreceptors that are going to sense the fact that the food contents coming in are probably going to be at a higher pH than the contents of the stomach itself, because the stomach is quite acidic, especially as we start producing the hydrochloric acid. Now, as this happens, this is going to send sort of a feedback loop. We're going to have more production of mucus, enzymes, hydrochloric acid, and the G cells will continue to produce gastrin, which will continue in a feedback loop to stimulate the production of these things. And that's going to allow for the contents of the stomach to be mixed back and forth, to be exposed to enzymes, to proteases, and a little bit to a gastric lipase, but that's not nearly as important as other lipases that we see along the digestive tract. But the main enzyme here is pepsin, and it's going to be working on proteins. Now remember, your salivary amylase and your lingual lipase, those enzymes can actually stay active to relatively low pHs, but eventually they'll be shut down by the low pH in the stomach, and pepsinogen will become very active because it is an enzyme that works at very low pH, and the contents of the stomach can reach 2 and sometimes even 1.5. So as this gastric phase continues, we're going to have the mixing of this material till it becomes a really nice, well-mixed, soupy chyme. And then we're going to have sort of waves of pressure that are going to be building in the pyloric region, and that's going to signal the pyloric sphincter to open up. And the pyloric sphincter will then allow for this soupy chyme to come down into the duodenum, and then we're going to have some sensations in the duodenum that are then going to signal back to the stomach to say, hey, okay, I got enough now, you can shut down. So we're going to have decreased pH, presence of lipids and carbohydrates that came from this mass in here that are going to cause the duodenum to do several things. First of all, it's going to release cholecystokinin and gastric inhibitory peptide, as well as secretin. And secretin and cholecystokinin are going to increase the secretions of accessory organs like the pancreas and the liver. At the same time, these signals are going to send a signal to the stomach to decrease secretion of enzymes and acids in the stomach, as well as to decrease the motility, so to decrease the amount of peristalsis. And at this point, we're going to, with cholecystokinin, we're going to signal the pancreas to start secreting pancreatic amylase and pancreatic lipase, as well as there are also some pancreatic proteases that are made, but pancreatic amylase, pancreatic lipase will continue the work of digestion of the carbohydrates and lipids that was started in the mouth, but then kind of shut down as the stomach got to a really low pH. So that will be picked up again in the duodenum. We will also see that it will stimulate the secretion of bicarbonate ion, which is going to be a very important buffer, which is going to help to bring the pH levels up within the duodenum. Because while the stomach is very, very well protected against hydrochloric acid, the duodenum not as much. It has some protection, but not as much as the stomach does. So we need to raise the pH in here to a more neutral level. And the other thing we're going to see is secretin is going to increase the production of bile in the liver. Cholecystokinin is going to not only increase the secretions by the pancreas, but also cause contractions of the gallbladder to push bile into the duodenum, and it's going to relax or open up a sphincter called the hepatopancreatic sphincter. And this sphincter is going to allow the secretions both from the pancreas and from the gallbladder to reach the duodenum. Now remember, we've got lipids coming in, so we're going to need to break those lipids down. So bile is important here because lipids are globs that do not react well with water. Lipids and water don't play well. So we've got to break down the lipids into smaller drops of lipids and even into their individual micelles, where we have a single layer of lipid with the hydrophilic heads toward the outside and the hydrophobic tails on the inside. We've got to break these large globs of lipid into the smallest droplets of lipid that we can so that we can give these lipases access to them, so the lipases can actually start to work on them. So that is an important function of bile, and cholecystokinin is going to open the hepatopancreatic sphincter and cause contractions of the gallbladder in order to put bile into the duodenum. And our secretin is going to increase the secretion of bile by the liver. Now, one of the things we've just talked about is the fact that our salivary amylase and lingual lipase kind of got shut down in the stomach. As the pH lowered in the stomach, they kind of shut down, and then the activity of pepsin started to kick up. So now we've got our protein digestion. The preliminary digestion of protein, in fact, is started in the stomach by the enzyme pepsin. And when we get to the duodenum, then we've got to pick back up the digestion of lipids and carbohydrates, and we do this with pancreatic lipase and pancreatic amylase. And so the stomach, when you consider what the stomach is doing at the height of its peristalsis in the gastric phase, our pH gets very low. And pepsin is one of these enzymes that is created by the proenzyme pepsinogen that is activated by extremely low pH. And the reason for this, obviously, is pepsin is not secreted directly by these cells, but secreted as the proenzyme, so the enzyme doesn't eat up the cell on the inside. So pepsin is going to be actually an enzyme that's very tightly folded in the beginning. So when it's pepsinogen, the precursor enzyme, I should say, the proenzyme, is actually tightly folded. And when it gets into the lumen of the stomach and that pH gets low, then it's going to start to unfold. And that's actually going to expose the cleavage site. It's like the pin on the grenade. You pull the pin out, now the grenade's ready to go. It's just like it cleaves off part of the peptide, and now the enzyme is in its active state, and it can get to work on those proteins. It's important to know that because of the harsh environment of the stomach, we really can't absorb much there. The stomach has to protect itself with its thick mucus coat. If that mucus coat is damaged, then the hydrochloric acid and some of these digestive enzymes, pepsin, can get in there and start damaging the epithelium of the stomach. And so, therefore, the stomach has to keep this thick coat of mucus, and if that coat is damaged, then you can start getting ulcers. And another thing to be aware of is if you push fluid from the stomach into the duodenum too quickly before the duodenum has time to prepare and produce enough bicarbonate, then you can get duodenal ulcers as well. So it's important to know that digestion of proteins begins in the stomach, but the absorption of nutrients won't really occur until later on, until we get into the small intestine. And most of the absorption of nutrients will be in an area called the jejunum.